Assalamualaikum and hi. Today my group will be reviewing an article about kinetics and modeling of kojic acid production by Aspergillus flavus in batch fermentation and resuspended micellar system. There are three objectives in this article. The first objective is to study the kinetics of growth, substrate consumption and kojic acid production by Aspergillus flavus linked strain 44.1. The second one is to develop an unstructured kinetic model based on logistic and Ludekin-Pyrrha equations. And lastly, to investigate kojic acid production in batch fermentation and resuspended micellar system. Kojic acid is a microbial metabolite with important applications in the medical, food and cosmetic industries. It is known for its antibacterial, anti-inflammatory, skin whitening, and anti-melanosis properties. It is mainly produced by Aspergillus species, particularly Aspergillus flavus link. Production can be carried out using either batch fermentation or resuspended micellar systems. Although culture conditions have been widely studied, there is limited information on the kinetics of kojic acid production and on mathematical modeling of the process. Therefore, this study proposes an unstructured kinetic model based on logistic and Ludekin-Pyrrha equations to describe growth, substrate consumption, and kojic acid production by A flavus link in both batch fermentation and resuspended cell systems. This model will help us better understand and control kojic acid production.
I'm Anis and now I'll be explaining about materials and methods. In the study, Aspergillus flavus link strain 441 was selected as the microorganism for kojic acid production. This strain is known for its high productivity and stable performance, making it suitable for fermentation studies. The strain was obtained from a reliable culture collection to ensure consistency and reproducibility. Selecting a high-producing and well-characterized strain is important because it directly influences fermentation efficiency and the reliability of kinetic analysis. Next, the culture medium was formulated to support both fungal growth and kojic acid production. Glucose at 100 grams per liter was used as the primary carbon source. This extract provided nitrogen and essential growth factors, while mineral salts such as potassium dihydrogen phosphate and magnesium sulfate supported enzymatic and metabolic functions. Ethanol was added to enhance kojic acid production. Optimizing the medium composition is essential because nutrient availability strongly affects metabolic pathway and product yield. For inoculum preparation, spores were cultivated on solid medium at 30 degrees Celsius to ensure high viability. A concentrated spore suspension was prepared and 10% volume-to-volume inoculum was used in all experiments to maintain consistency. Batch fermentation was carried out in two systems, which is shake flask, which provided uncontrolled oxygen condition, and a stirred tank fermenter where oxygen supply was controlled. Comparing this system allowed evaluation of the effects of aeration and mixing on fermentation performance. In the stirred tank fermenter, dissolved oxygen was controlled using a two-phase strategy. During the growth phase, oxygen saturation was maintained at 80% to promote biomass formation. During the production phase, oxygen was reduced to 30% to favor kojic acid synthesis, while agitation was kept constant at 600 rpm. Additionally, a resuspended cell system was used. Cells were harvested at stationary phase, washed and resuspended in a glucose solution without nitrogen. This method separates growth from production and allows more efficient substrate conversion. Several analytical methods were applied to support kinetic modeling. Cell growth was measured as dry cell weight to accurately represent biomass concentration. Kojic acid concentration was determined using HPLC at 265 nanometer for high specificity and accuracy. Glucose concentration was measured using the DNS method, while nitrogen content was analyzed using the JERDA method. This measurement provided the experimental data required for model development and validation. Then, kinetic modeling was used to mathematically describe fermentation behavior. Cell growth was modeled using the logistic growth model, which accounts for the showing of growth as nutrients become limited and the culture approaches the stationary phase. Product formation and substrate consumption was described using the Ludekin-Pyrrha model. In this model, the parameter alpha represents growth-associated product formation, while beta represents non-growth-associated formation. This concept helps identify whether kojic acid production mainly occurs during active growth or continues after growth has stopped. Lastly, for data analysis and model validation, model parameters were estimated using nonlinear regression in MATLAB. Experimental data were fitted to the proposed kinetic models and predicted value were compared with measured results. Statistical analysis shows good agreement between experimental and predicted data, confirming the reliability of the models. This validation demonstrates that the kinetic models can be used to support process optimization and scale up.
Hi everyone, I'm Anyi and I will be summarizing the result for kojic acid production in batch fermentation. So in this study, the kojic acid was produced using two different systems, a shake flask and a fermenter. From the results, we can see that the biomass concentration were quite similar in both systems, but slightly higher in the fermenter. The maximum biomass in the shake flask was about 11.8 grams per liter, while the fermenter reached about 12.1 grams per liter. However, the difference in kojic acid production was much more significant. In the fermenter, the maximum kojic acid concentration reached 36.5 grams per liter, which was clearly higher compared to the shake flask. This higher production also resulted in better yield and overall productivity. So one of the main reasons for this improvement is likely due to aeration. Shake flasks have limited oxygen transfer, while the fermenter provides better control of oxygen availability. So these two figures 1 and 2, show the experimental and calculated data for both systems. The simulation curve fitted very well to the experimental results with more than 93% confidence. Only slight deviation was observed at the beginning of the fermentation, and statistical analysis using a t-test confirmed that the difference between calculated and experimental data were not significant at 5% probability. This means that the proposed kinetic models based on the logistic equation and the Ludekin-Pyrrhot model are reliable in describing the cell growth, glucose consumption, and Kojic acid formation by air flavus link. So Table 1 presents the kinetic parameters for both systems. Even though maximum biomass concentrations were similar, the maximum specific growth rate and the non-growth associated rate constant for Kojic acid formation were noticeably higher in the fermenter. In fact, the specific growth rate was about 3 times higher and the non-growth associated constant was about 2 times higher than the shake flask. Interestingly, the value of M was 0 for both systems, indicating that the Kojic acid production is non-growth associated. Another important observation is related to glucose consumption in the shake flask. More glucose was consumed during the growth, leaving only about 59.6 gram per liter in the culture after the stationary phase, compared to 77.6 gram per liter in the fermenter. This lower remaining glucose supply in the shake flask may have result in limited Kojic acid production. Finally, the kinetic study shows that the Kojic acid fermentation occurs in 2 phases, a growth phase and a production phase. During growth, the glucose and nitrogen are used to build biomass and enzyme systems. Once cells stop growing, the remaining glucose is then converted to Kojic acid. The production stops once the glucose is depleted, even the cells are still metabolically active, and the Kojic acid levels start to decline due to glucose depletion and due to the degradation into oxalic acid and acetic acids. Overall, its findings show that the fermentation in the controlled environment of a fermenter significantly improved the Kojic acid production, supported by the strong kinetic model validation.
Hello everyone, I'm Pang, and here I'll be summarizing the result of Kojic acid production in a resuspended cell system. The graph here shows a typical time course of Kojic acid production in a resuspended cell system in a shake flask culture. The graph plots incubation time in hour on the x-axis with Kojic acid, which is the product concentration, and glucose, which is the substrate concentration, on the y-axis. The graph also shows the fitness of the calculated data according to the kinetic models for a resuspended cell system shown there. The calculated data fit very well to the experimental data, and deviations are not significant at a probability of 5%. In this graph, there's a point to take note of. We can see that cell concentration remains constant at a 12.2 gram per liter throughout the process. This is because there's no addition of nitrogen source to the resuspended cell system for Kojic acid production, and hence cell growth does not occur. So in the graph, we can see that Kojic acid production increased while glucose concentration decreased linearly with incubation time, then stopped when glucose is depleted. The kinetic parameter value of N and beta are similar to the values from the batch fermentation in a shake flask, and so the yield and overall production of the resuspended cell system is 0.38 and 0.069. And next here, we are looking at 2 graphs showing the effect of 4 different cell concentrations on Kojic acid production and glucose consumption in a resuspended cell system. As we can see from the graphs, in all cases, Kojic acid production increased and glucose concentration decreased linearly with incubation time. In a paper, it is mentioned that the experimental data plotted in the graph are compared to the model prediction showed as solid line, and the t-test was performed to check whether the deviations are statistically significant. And it is found that for cell concentration of 3.36, there's a significant deviation after around 800 hours, which might be due to slight enzyme deactivation during prolonged incubation. And here, table 2 summarizes the 4 cell concentrations and lists the kinetic parameters. From the table, we can see that the value of N is about 0.119 to 0.0128 at lower cell concentration, then fall to 0.0098, and then fall to even lower at the higher cell concentration. The glucose consumption constant beta follows the same trend. This is due to the increased viscosity of the reaction mixture, leading to decreased mixing efficiency and cell concentration increase. Enzyme action is reduced and the conversion rate of glucose to Kojic acid is decreased. The Pmax increased with increasing cell concentration, and the process time shortens as well as concentration increase, leading to a proportional increase in overall productivity. In short, the graphs and table provide that resuspended mycelia produce Kojic acid in a substrate-driven and non-growth manner with linear kinetics where glucose is available. Increasing cell concentration is an effective way to increase productivity, same as using very high glucose concentration.
I'm Ishira. I will be concluding our study on the kinetics and modeling of Kojic acid production. So to conclude, this study demonstrated that Kojic acid production by Aspergillus flavus can be effectively described using logistic and Ludekin-Pyrrhot kinetic models. The results showed that the fermentation performance is highly influenced by the oxygen availability, with the fermented system producing higher Kojic acid yields compared to the shake flask. It was also confirmed that Kojic acid production is a non-growth associated process, occurring mainly after the cell growth has stopped. And meanwhile, in the resuspended cell system, the efficient and stable production was achieved even without further cell growth, especially at higher cell concentrations. So overall, this study highlights the importance of controlled fermentation conditions and kinetic modeling in improving and optimizing Kojic acid production for industrial applications. These are our comments on the journal. So overall, the study demonstrates that the kinetic modeling using logistic and Ludekin-Pyrrhot equations effectively supports process optimization by quantifying the effects of key parameters, comparing production systems, predicting process behavior with high confidence, and guiding the strategies to improve yield, productivity, and industrial efficiency.